Welcome to this lecture on the biochemistry of metabolic processes. Today, we are going to explore a fascinating and crucial metabolic network known as the pentose phosphate pathway. While you might already be familiar with how cells generate energy, this pathway reveals a completely different side of cellular survival and growth. It is a beautiful example of how our bodies dynamically adapt to meet immediate biological needs. Let us dive in and understand how this works. To understand this pathway, imagine you are driving down a massive, busy highway whose only purpose is to get you to your destination as fast as possible. In the biological cell, that main highway is glycolysis, which breaks down glucose to create raw energy in the form of ATP. But what if you do not need energy right now? What if instead you need building materials to construct a new car or protective gear to survive a storm? That is exactly where the pentose phosphate pathway comes in. It acts as a vital metabolic detour. It is a specialized side road that glucose takes when the cell needs specific molecular tools rather than just raw energy, willingly sacrificing some potential ATP to gather essential supplies for growth and protection. Now that we know why this detour exists, let us look at how it is structured. The pentose phosphate pathway provides an alternative metabolic route for the breakdown of glucose 6-phosphate. Instead of splitting the sugar to extract energy, this pathway is divided into two distinct phases that serve different cellular needs. The first is the oxidative phase, which is strictly irreversible and is responsible for generating protective molecules. The second is the non-oxidative phase, which is reversible and acts like a recycling center, interconverting various sugar molecules. Depending on what the cell needs at any given moment, it can dynamically shift the flow of molecules between these two phases. This incredible flexibility makes it one of the most versatile networks in all of biochemistry. This dynamic shifting brings us to a very important question. Why go out of the way to use this pathway? The answer lies in survival. The first major product of this pathway is NADPH. You can think of NADPH as a powerful reducing agent that acts like a chemical shield for the cell. It actively protects cells from dangerous oxidative stress and free radicals. Without this vital shield, fragile cells like red blood cells would literally burst open under the stress of oxygen radicals, leading to a severe and life-threatening condition known as hemolytic anemia. So producing NADPH is absolutely non-negotiable for cellular defense. Beyond just protection, this pathway is also fundamentally about growth. The second major product generated here is ribose 5-phosphate. This specific 5-carbon sugar is incredibly important because it forms the structural backbone of both DNA and RNA. This means that any actively dividing tissue in your body relies heavily on this exact pathway to supply the raw materials required to write new genetic code. Whether it is a skin cell repairing a scratch or an immune cell multiplying to fight an infection, they all depend on this metabolic detour to provide the building blocks for their genetic material. Now that we understand the biological purpose, let us look at the exact chemistry that makes it happen. This formula represents the irreversible oxidative phase. It starts with glucose 6-phosphate, our 6-carbon sugar derived from our diet. The symbol NOx represents the empty electron carrier molecules, NADP+, waiting to be loaded. As water is consumed, one carbon atom is completely removed and exhaled as carbon dioxide. The remaining 5-carbon sugar is ribulose 5-phosphate, which later becomes the ribose for DNA. Most importantly, this single cycle produces 2 molecules of N-red, which are the fully loaded NADPH electron carriers. If the cellular concentration of empty carriers increases, it physically signals that the cell is running out of protective power, which speeds up this entire reaction to compensate. Let us apply this chemical equation to a real biological scenario. Suppose a rapidly dividing skin cell is preparing to replicate its DNA and needs to synthesize exactly 150 molecules of ribose 5-phosphate to build new nucleotides. We need to determine how many molecules of glucose 6-phosphate must enter the pathway. Because the biological ratio of the starting 6-carbon sugar to the final 5-carbon sugar is strictly 1:1, there's no complex multiplication factor needed for the primary carbon backbone. We simply multiply the target of 150 by 1. The cell must commit exactly 150 molecules of glucose 6-phosphate to the pathway to meet its goal. If the cell needed more ribose, this input requirement would increase in direct linear proportion. Following up on that exact same scenario, we must also calculate the byproducts generated during this process. We know from our stoichiometry that each cycle produces exactly 2 molecules of NADPH and 1 molecule of carbon dioxide. To find the total NADPH, we multiply our 150 cycles by 2, giving us 300 molecules. This massive stockpile of reducing power is a highly beneficial side effect of preparing for cellular division. Finally, since exactly 1 carbon atom is lost as a gas molecule per cycle, we multiply the cycles by 1 to find the carbon dioxide exhaust. The final result is 300 molecules of protective NADPH and 150 molecules of carbon dioxide, beautifully illustrating how deeply interconnected these metabolic pathways are. This interconnectedness leads us to another critical calculation, determining the metabolic cost of cellular defense. Sometimes a cell is under attack and needs NADPH immediately. In this formula, we work backward. The numerator represents the total biological demand of NADPH needed to fight off oxidative stress. The denominator represents the fixed biological conversion rate, which, as we saw earlier, is 2 NADPH molecules produced for every 1 glucose 6-phosphate molecule consumed. By dividing the total required protective molecules by the yield per cycle, we isolate our unknown input variable. This tells us exactly how many glucose molecules must be sacrificed, giving up their potential ATP energy to save the cell from destruction. If the oxidative stress increases, the demand for glucose diversion increases proportionally. Let us see this defensive calculation in action. Imagine a red blood cell is suddenly exposed to a high level of oxidative stress from a malarial drug, causing a dangerous buildup of hydrogen peroxide. To neutralize this lethal threat, the cell's defense system requires exactly 500 molecules of NADPH immediately. We need to calculate how much glucose must be diverted away from energy production. Using our formula, we take the biological demand of 500 and divide it by our fixed yield of 2. The calculation reveals that the cell must divert exactly 250 molecules of glucose 6-phosphate into the pentose phosphate pathway. This physical interpretation highlights a critical biological trade-off. The cell willingly gives up potential energy to prioritize its immediate survival. Now that we have calculated the costs and yields, let us look at the entire pathway visually. On the left side of this schematic, you can see the irreversible oxidative phase. Notice the starting molecule, glucose 6-phosphate, and follow the arrows downward. You can clearly see the crucial moments where NADP+ is converted into our protective NADPH, alongside the release of carbon dioxide. This leads us to ribulose 5-phosphate. Moving to the right side, we enter the non-oxidative phase. Notice how the arrows here are double-headed, indicating that these reactions are reversible. Enzymes like transketolase and transaldolase shuffle carbon atoms around, eventually creating fructose 6-phosphate and glyceraldehyde 3-phosphate. The arrows pointing outward show how these molecules can seamlessly re-enter the main glycolysis highway if needed. This diagram perfectly captures the dynamic, interconnected nature of cellular metabolism. As we wrap up this topic, let us review the most important ideas we have covered. The pentose phosphate pathway is not just a background process. It is a vital metabolic detour that prioritizes cellular building and protection over raw energy production. By oxidizing glucose 6-phosphate, it provides the essential ribose 5-phosphate, necessary for synthesizing DNA and RNA, ensuring cells can grow and divide properly. Simultaneously, it generates NADPH, a crucial reducing agent that defends our cells against lethal oxidative stress. Understanding this pathway reveals the incredible adaptability of cellular metabolism in real time. You have now seen how our bodies constantly and beautifully balance the immediate need for energy with the long-term need for structural integrity and defense. Great job mastering this complex topic.